This is the fourth and final video in this series. We saw in part one of the series that evolution is not true. When we applied the scientific method to evolution theory, we found that the predictions that evolution theory made are untrue, and therefore the theory itself is not only not true, it's not a science. In part two, we saw why biblical creation as a history in chapter 1 of Genesis is true. We saw strong scientific evidence that supports it. In fact, it cleared up some complexities, some problems with the interpretation of the fossil data for evolution theory. In any event, evolution is not true. Historical biblical creation is true. Then in part 3, we went a little bit beyond in the book of Genesis and found that genetic test data now that are emerging have found the timing of when Adam and Eve and Noah first existed. And we end up today in the fourth and final part where we get into another part of Genesis, and that is the 1,000-year lifetimes and the development of the human races. Now, in the book of Genesis, we know that Adam lived about 930 years, and Methuselah 969, and Noah 950. Most of the people within that time span lived in excess of 900 years, with I think the earliest passing away at about 360 years. So extremely long lifetimes. And the question we ask in this particular part is there scientific support for the nearly 1,000-year lifetimes? Is there a known scientific way that people could have lived as long as 1,000 years, whereas we now live with a maximum age of approximately 120 years, as it says in the Bible and as we see in evidence around us? So in order to answer this question, what we need to do is to look at something called telomeres. If you remember from part 1 and part 2 and part 3, when we talked about some of the aspects of DNA, you saw that the tail ends of the chromosomes in DNA are what we call telomeres. This was a discovery that happened about 10 years ago, and in 2009 the Nobel Prize was awarded for the understanding of the extreme importance of these things called telomeres. So let's look a bit at that. Now here again is the same picture that we saw, where we have a very simplified chromosome. It's got these genetic material, these genes in it, and then these separator regions that separate genes, with the genes being the things that determine all of the characteristics of you, your body, and all of its parts. At the ends of each one of these chromosomes, we have some material called telomeres. Now these telomeres, after the development of the human genome, so-called, after the genetic structure and the specific genetic constitution of chromosomes was mapped in the mid-90s during the Clinton presidency, it was believed that these tail ends that we now call telomeres were just called junk DNA. They appeared to provide no benefit. They had no purpose. They were just drags and tail ends of chromosomes that seemed to have no importance and no meaning. Well, in actual fact, the discovery of the importance of these ends and the naming of them telomeres is analogous to the situation. Suppose we've all had a shoelace, and your shoelace has these little plastic cylindrical things that have been given the name aglets, and the telomeres perform the same thing for chromosomes that these aglets perform for shoelaces. We all know that if we want to lace up our shoes and those aglets are absent and the ends of our shoelaces have frayed, they have a difficult time lacing them up. It's important to have those aglets present. What do we do usually? Well, we might do something real specific to add tape around it or something, repair it for a while, but it will ultimately usually we replace the shoelace to get one with new aglets so we can more easily lace up our shoes. Well, telomeres perform that same function for chromosomes in that they are, in some sense, sort of sacrificial lambs, sacrificial ends of these chromosomes that, as long as they're there and they're of a certain length, they protect the integrity of the chromosomes. And that's a very important function for these telomeres to perform. They're no longer junk DNA. Now, in terms of the structure of where these telomeres are, here we have again, we've seen in prior videos, the cell with its nucleus. And inside of this nucleus, we have chromosomes. This is just one part of a very simple structure of a chromosome. At the tail ends, again, are telomeres. There is also within the nucleus of the cell an enzyme that was discovered at the time that the importance of these telomeres was determined. It's called telomerase. And this enzyme has as its function to protect, promote, and enhance the length of these telomeres so as to protect the integrity of the chromosome. So its main function is the protection function of telomeres. And the telomerase is something that aids in their maintenance. It's something that was in that cell to try to prevent the reduced length of these telomeres and in fact to maintain it and promote it. The problem is that telomerase is in a finite small quantity and it is not renewed. It behaves like a counter for the maximum number of cell copies. As you need new cells in your stomach or any other part of your body every 2 or 3 days in the stomach, every 25 years in your bones, the first step that was made, if you remember, is that we make a copy of this DNA. Well, we try to make a copy of the entire thing, but the concentration is in the copy of the central part here where the genes are. And these outer bands, these telomeres, are not copied very accurately. Well, if they're not copied very accurately and they're shortened, then this telomerase performs its function for a while until it's all used up to continue to support that telomeres, to enhance their length or at least to prevent their reduction. But once the telomerase is all gone, then the cells on their duplication and the duplication of the DNA is on its own in a sense. Now, the thing that we know about this telomerase enzyme that's in cells is that the gene that makes it is turned off. We make no more telomerase in our cells, and therefore we have not only a finite life, but that life is determined by the amount of telomerase initially presented and the maximum number of copies of cells that we can make. So that's the general situation with telomeres and telomerase within each and every one of your cells. Now, here is what is known at this point in terms of what's going on with these telomeres and how do they shorten with age. We know that they shorten with age. Older people have shorter telomere lengths. Younger people have longer telomere lengths. And in fact, this telomere shortening is something that we have known all along. It's called aging. It's when your cells can no longer accurately reproduce and we have difficulties, we have diseases, we have problems, and ultimately we die because our telomeres are all gone more or less and our cells can no longer accurately reproduce, say in the kidney for example, or some of the heart cells or whatever, and these organs dissipate and ultimately die, and the whole being, you and I, will ultimately die. Now, at time of conception, the ovum and the sperm have, it is known, about 15,000 what they call base pairs, but in effect 15,000 beads at the end of each one of those chromosomes, 15,000 pairs of each of the beads on the two strings. In other words, I call them base pairs, or two beads that are at the same location in the opposite strands of the DNA. So you have about 15,000 at conception. During the 9 months of rapid reproduction of cells and growth of the fetus inside of the mother, it's down to about 10,000 at birth. So at birth, babies typically have about 10,000 telomeres in their lengths at the end of their chromosomes. And here are two sets of data that are out there in the literature. They certainly are not the final answer here, but they show the same general trend that as people get older, their telomeres on the average are shorter. Now, you can have a commercial test done as an additive for your blood test. It costs somewhere in the $300 to $400 range, and it's probably not covered by hardly anybody's insurance. but you could get your average telomere measured. Suppose you had done such a test and you ended up with a result, and it would be compared to this curve. You would end up a result, say, up here around 7,000. What that would mean that this person at the age of about 70 has the biological body of about a 30-year-old. Good news, long telomeres, long expected life, at least from the point of view of natural causes, and you'll see in a minute, less potential for having disease because the cell duplications are more accurate for a longer time. On the other hand, if your test happened to be substantially below the line, that would indicate you have not only short telomeres, but you could potentially have a disease problem, and your life expectancy, even at 70, would probably not be much longer than that. Now, they also believe that if that telomere length gets below about 3,000, that is a significant problem. That's going to lead, as we'll see in the next slide, to not only shorter lives, but being prone to diseases because your cells are not accurately duplicated. Now, a good news, if you are interested in prolonging your life or having the chance of prolonging your life or minimizing diseases, there has been at least one supplement that has been discovered based upon this research. It's called TA65, and it's an extract from the astragalus plant. That is a plant that the Chinese have been using in combination with other herbs over time to treat various maladies. But this particular TA65 uses a very highly purified, and they talk about it being a single molecule of this astragalus plant that enters into your body if you take this material, and it promotes telomere lengthening. It costs about $8,000 per year, so it's very expensive, and I'm not sure that we all are interested in knowing exactly where we stand on this curve, if our projected lifetime in the beyond the test is going to be short, or if we can go jump out of balloons or something and skydive and all that because we have such a young body. But in any event, this is a situation with telomeres that are known, and it's a developing area of technology, I would say, that certainly with the next few years to a decade, we're going to know this a lot better, and it will be something that you could take advantage of if you really wanted to know. Now, the maximum lifetimes of 120 years that currently we observe, we know that there occasionally is a man or a woman above 120, 124. Recently, I think, was the oldest person who passed away, but it's typically 90 to 100 years is the maximum, but if you live to way up to 120 years. What we know also in studies of genetic material is that there is a gene within DNA that has as its ability to make more telomerase. This gene can make more telomerase and therefore protect your cells longer and therefore give you a longer life, but we know that that gene is now turned off. That gene is turned off. We are not making any more telomerase other than we initially had in our cells when we were born, and therefore our lifespan is particularly limited to, as we see, around 100 to 120 years. Well, it's not too easy to figure out that, well, if that gene had been turned on, if it were turned on in the past, that lifetimes could have been significantly longer, perhaps 1,000 years. This does not prove that telomeres and telomerase are the way that the early people reported in the Bible from Adam and Eve through Noah and beyond had their telomerase gene turned on, but it certainly is a scientific explanation for how this could have happened. It is not unscientific to think that people could have lived in the past as long as 1,000 years. We have here a scientific way in which it can be explained. Now, as I said earlier, long telomeres are the goal. They correlate with longer lifetimes. Your cells reproduce more accurately, or at least the genetic material within your chromosomes and within your cells copy more accurately, and therefore your body functions better for a longer time. And it's also been found that that correlates with minimal diseases. Even cancers and all the other diseases that we see in ourselves and other people correlate with having short telomeres, such that short telomeres, as we said, give shorter lifetimes, and people with short telomeres have many more diseases. So that's what is currently known about telomeres and telomerase, and how it relates to the possibility that people could have lived as long as 1,000 years, and gives us some idea what we could do about this in the future should we choose to take that supplement TA65 or do other things. In fact, there is a book upon which this information was reported called The Immortality Edge. It's in the references on the website, and the three authors of this Immortality Edge have written a book showing how if you take this TA65, and all three of them are taking it all the time, and how you couple that with a good diet and exercise, that they potentially are themselves going to be some of the first experiments to see whether longer lifetimes and healthier lifetimes are the result of doing that. Now, there is a caution for living too long. If one were to live 1,000 years, or had the potential to live 1,000 years, you would quite likely would die before that from cancer, even though one had long telomeres. And that is because astronomers and cosmologists know that there was a supernova called Vela that exploded about anywhere from 12,000 to 50,000 years ago. They don't know, a little bit of controversy exactly when this happened, but what happened was a supernova, which is a very large star that burns out in its interior too fast, and when its interior is burned out too much, the outer layers of the star by gravity come flying in and imploding on the center of the star, and the net result is that a whole bunch of cosmic radiation is released. Now, this is the closest, by far, the closest supernova that has occurred in terms of distance from Earth, about 800 light years away. And the net result of that supernova Vela exploding tens of thousands of years ago is that it increased the cosmic radiation on the surface of the Earth by a factor of 30. This may be coincident with the Tower of Babel dispersion time in the 12 to 50,000 year range, or anyway, sometimes after the time of Noah. Don't know exactly anything more about that. But this dramatically increased level of radiation would likely result in cancer if a person lives such a long time. Cosmic rays are a known carcinogen, and the current level, if you live a substantially longer life, you will quite likely get cancer from that incident radiation that comes into your body and can disrupt individual atoms or molecules within your body and lead to cancerous problems. So that's a caution anyway for living such a long time and not necessarily being healthy in the presence of this high level of cosmic radiation. Let's move on then to the last area, and that is human races. Where do these human races come from? And we'll look for explanations for that. The human races mainly differ in skin color. There are other attributes of the different races that we see here on Earth currently, such as stature and individual body part structures, eyes, etc., arms. But they all have essentially the same DNA. We do know that they have the same DNA. And so in effect, or in truth, all humans are the same species, the same. The biblical explanation is that at the time of the Tower of Babel, now this was after the time of Noah for a bit of time, in which humans were again getting into all form of evil, turning to the dark side, and God decided again to do something about this, not to send another flood, but to disperse the humans to different locations on Earth. It says that God dispersed all different peoples around the different positions on the Earth, and in order to further confound their communication with each other, he gave them new languages. So the biblical explanation is that the races are a result of dispersion to different parts of the Earth, and we'll see scientifically how that is consistent with something we know. On the other hand, evolution theory uh, believes that there was a continental rift, a continental volcanic plate type altercation that occurred in Africa that resulted in a drought. And because there was a drought, there was nothing much to eat, and so that forced humans to migrate to different locations on Earth where they could not have a drought and not have that rift present. And so that's the explanation there. Now, evolution is very careful to say that the races did not result from different evolutionary paths, with the idea that evolution is an unplanned, small time step, long time span phenomenon. It would be highly unusual to end up with 4 or 5 different human species, different types of humans having evolved separately in different parts of the Earth. And so that's a point that is made that certainly these are not different species that evolved separately, because it'd be very unlikely for 5 very similar ones to have evolved separately. And it's also unlikely that they resulted just by chance. So that's really saying the same thing. Now, in order to look at the scientific explanation for where the human races might have come from, we have to look at 2 things called adaptation and vitamin D. Adaptation and vitamin D. So let's look at adaptation first. Now, adaptation and mutation are things that are put together usually. And adaptation is a change in a given species of plant or animal that is an inherent characteristic of that plant or animal that allows it to change itself a little bit to more comfortably deal with the environment that it is placed in. And so adaptation is an inherent flexibility, if you will, within all plants and animals that allows them to alter their characteristics a little bit to take advantage of certain genetic possibilities within their DNA to more readily live a good life in a given location on Earth. So adaptation is something that's common to both evolution ideas and to God's plan for setting up humans and all other species such that they would not be so rigidly able to withstand only one set of climatic conditions or earth conditions, but would be able to adapt. Now, mutation, on the other hand, is something that can occur as a result of genetic material altering its characteristics, say during conception or by some other means during the lifetime of an individual, where some genetic mutations can pop up. So these 2 things, adaptation and mutation, are things that can alter the outward characteristics of certain living beings. And so we have the ability to adapt, whether it's from climatic or environmental conditions. Evolutionists call this microevolution. Macroevolution is the one where one species evolves into another species, and it's the macroevolution that is not true in terms of what we've seen in these videos, in terms of what I believe as a result of studying this area pretty extensively. But microevolution is just these small changes. The most famous of these appears in Darwin's original On the Origin of Species, his original book in 1859, where he was in the Galapagos Islands and he observed on the various islands a bird that seemed to be the same bird, which was a finch, but he found that there were variations in those finches. And in particular, on an island where there were large seeds, there were larger beaks in these finches. This large beak here can more easily crack a nice big seed, whereas on other islands where there were smaller seeds, the birds had smaller beaks, such as this same finch, but has a smaller beak. It doesn't need a bigger beak. And he thought that that was interesting, that that was at least an indication that evolution could potentially be true. He thought he might be seeing an example of how evolution might work. But in actual fact, he himself agreed that this and other things he looked at, such as selective breeding of horses and cattle to end up with a certain characteristic in a given species, that was not really evolution. When you got all through, the evolution was really microevolution, so-called, and the finch was still a finch. Its DNA was no different. It just had adapted to the needs on a particular location where it was living. And in fact, since the time of Darwin, it has been shown that this is reversible. That when small seeds are become available, that the beaks become smaller again. No need for a big, large beak. So this is adaptation in a case, and one of the examples that's brought up quite often in evolution texts is that of the woolly mammoth. The woolly mammoth is a big elephant-like creature, lived in various places, but actually in the northwestern United States they find mammoth bones, a large being that when it became extinct, it had lots of hair on it. They think that there were prior versions of that mammoth that were not woolly, didn't have hair, and the general idea was here is that when there was a mild climate, you didn't need hair to stay warm. But when it went through a very cold climatic time, that then those particular animals that had a bit of hair are the ones that prospered and ultimately adapted to have most of the species have lots of hair and therefore able to survive until they became extinct. The major point about this adaptation and mutation is that in the end, the species are still the same. No new species occurs. It's just alterations, adaptations, and in some cases mutations within a given species that allow it to adapt to the living conditions that it had. Darwin spends several chapters on these sorts of microevolution type things, but he himself and all evolutionists beyond that have concluded that these are not evidence that evolution of one species into another species can occur, but are just innate characteristics of living beings. Now, the other aspect that will explain the adaptation, the development of the human races is that with vitamin D. A strange thing to have just one single component in your body that's so important that it could have led to the adaptation, the development of the different races. Vitamin D, its importance was discovered in 1923 when a food services professor was studying rat food. Now, in his technical papers that he wrote about this, based on his discoveries, how to make rat food good, it's not said why he was studying rat food. And of course, the only logical thing is they were using lab rats at this time for various studies, and he was interested in developing a food that was good for these rats so that they were good and healthy when they were put into the testing that they went through. And what was found before this, that the lab rats that were being used end up with broken bones. That they had broken bones, and it wasn't until their food was irradiated with ultraviolet light that all of a sudden these lab rats, future generations, ended up having no bone problems. In actuality, we know that disease in humans, and we call it rickets. The rats were suffering from rickets because their food did not supply something that they needed. And they also were living inside and were not accessible to sunlight. And that is the reason why they developed rickets and they were deficient in vitamin D. We know that vitamin D is generated within the bodies of most mammals by sunlight. We'll see that in more detail in the next few slides. As a result of this discovery, studying the health of lab rats, since 1938, milk has been fortified with vitamin D to prevent rickets in predominantly dark-skinned children living in the higher latitudes. And we'll see how that makes sense in a bit. But basically, when black children were displaced one way or another from Africa, where they were normal environment, and moved to the northern latitudes, that these young black children developed rickets. Their bones would break more easily. And it was ultimately realized that they were not getting enough sunlight through their dark skin to make enough vitamin D to keep their bodies healthy. Therefore, milk was added with a vitamin D in order to provide this supplement, since most young children will drink milk. Now, vitamin D is naturally in only a few foods: fish oil, mackerel, salmon, tuna, eels, whale oil, and a few very more obscure substance contain natural vitamin D. So you can't get too much vitamin D by eating natural foods. You get it by sunlight, being outside with sunlight, and that's the other source. Currently, vitamins, of course, do contain vitamin D that some people take in order to achieve sufficient vitamin D. So vitamin D is crucially important not only for broken bones, but we'll see for other health aspects. Now we need the right amount of vitamin D. That's important. We need the right amount to live and be healthy. You get broken bones if there's not enough vitamin D, and if there's too much, you have organ problems and general ill health. So it needs to be the right amount of DNA, enough but not too much. And as I said, vitamin D is manufactured within, we'll see in a minute, in the lower skin layers from incident ultraviolet sunlight. And the main reason to have vitamin D, though that's not the only reason, is to process calcium. If you don't have enough vitamin D in your body and you take in calcium in various ways, say that's unfortified milk, you get the calcium, but your body cannot process that calcium for use, for example, in your bones. That's why rickets results from someone who cannot effectively use calcium because he does not have vitamin D in his body, enough vitamin D. Now we'll see in a minute that human skin color, or pigment in the skin, controls the vitamin D that's produced for a certain amount of incident sunlight. And the net result of this look at vitamin D is that the human races are the likely result, as you see below here, the human races are the likely result of adapting to different living locations on Earth with also some beneficial genetic mutations. It hasn't said much about the mutations, but that is certainly also a possibility to have occurred to result in part of the characteristics of the various races. But the dominant thing is this importance of vitamin D and how that works in bodies. So let's look at that a little bit more. Now, one thing we know, and we know pretty well for any particular location on Earth, exclusive of cloud cover and rain, etc., one can calculate how much radiation hits that particular location on Earth as a function of time. Say this is shown here for a year. So for 1 year, if you lived about on the equator at Nairobi, Kenya, you'd have about 10 units here of this, relatively obscure units, but kilowatt hours per square meter per day. But you'd have 10 units of sunlight, ultraviolet radiation that's coming to hit your skin. If you live far away from that, and far away you can get from the equator is the north or south pole, but if you live on the North Pole, you get barely a third of that amount of radiation on the North Pole. And so the amount of radiation, depending upon where you are on the surface of the Earth, varies rather significantly. Now let's look at a black-skinned person, a dark-skinned person living on the equator, living in, say, Kenya, Nairobi, Kenya. The first two layers of a person's skin are the ones where we have pigment, and here I've shown it as a dark pigmented skin. Here I've shown 10 rays of ultraviolet light that are coming into the skin as a result of that person living outside. And that person then is able to absorb a bunch of that radiation and result in only 2 out of 10, or 20% of that ultraviolet light reaching the 2 layers that make the vitamin D. These named layers here are the 2 that result in the manufacture of vitamin D that's used by the body. So only 20% of that potential ultraviolet light to make vitamin D gets through. But there's a lot of sunlight coming. There's a lot coming into this area, and so with that dark skin and only utilizing 20% of it, that's just the right amount of vitamin D in the body to lead to a healthy lifetime for a dark-skinned person. Now for a light-skinned person, let's look at a light-skinned person living on, say, the North Pole. With light skin, those same 10 rays of light coming through, 6 out of 10 get through. They're not absorbed. Only 4 out of 10 are absorbed in these upper outer skin layers. So a bunch of the radiation gets through, 60%. But with that location on Earth, there's not all that much sunlight that's coming. But 60% of a smaller number is just the right amount of energy to get through to make just the right amount of vitamin D in one's body. And so this is the explanation that as dark-skinned people from the time of Noah and the original time when all of humanity was concentrated, when they were dispersed to different locations on Earth, they, over a relatively short period of time, adapted to having lighter skin colors so as to make efficient use of the sunlight that was of a lesser quantity at the particular location they lived. Now it's very interesting, I mentioned earlier that the fish oil and various oils from sea life were a way to get vitamin D, and the only race living at a location on Earth that doesn't jive with this, what I've just told you. In other words, there is a dark-skinned population living at a high latitude on Earth, in particular the Eskimos, who get very little sunlight. In fact, you always see them in their parkas where most of their body is covered up. But the one thing we know that they live on quite a bit is fish oil. They hunt the various oil-producing species of sea life that give them sufficient vitamin D as a result of consuming fish oil. So this is rather an interesting area of this whole business where the color of one's skin is very important in order to, in terms of where you live. And if not, of course, these days, one can move to any location on Earth and have sufficient vitamin D. In particular, if white people are in equator in Africa, you have to be very sure that you get covered up to not get too much ultraviolet light. And in fact, we also know that what happens with a person who spends a lot of time out in the sunlight without having protection, they get a tan, and in some cases a very deep tan. This is the body's adaptation to limit the amount of vitamin D that is produced within these lower layers of the skin cells. Anyway, that's the effect of skin color, and vitamin D is the thing that has the scientific explanation for how these various skin colors could have developed over time, which results in the different races on Earth. So let's just conclude what is going on in this area. First of all, in thousand-year lifetimes, we've seen that it's feasible. That telomerase is the thing that could do it. All we'd have to do is turn this telomerase making gene on, and much longer lives would be possible. Currently, humanity has it turned off, so we only live approximately 100 or 120 years as our maximum age. Early humans could have had this on, and it could have been God's plan to have longer lifetimes, not the infinite lifetimes that probably were God's plan for Adam and Eve when they were first created, but to allow life ages as long as 1,000 years with this telomerase gene turned on. And we've just last seen that the human races could have been the result of adaptations to different climatic conditions resulting from the body's need for just the right amount of vitamin D. Since this is the end of the 4-part series, let me just very briefly go over the final conclusions, as I say for now, that have been reached as a result of looking at evolution theory and looking at biblical creation history. I think that these conclusions will change only slightly over time, but the numbers might change a little bit for some of the scientific things, but I think, will end up with the same general conclusions. In part 1, we saw that evolution theory is not true, that when we applied the scientific method to it, it was not only not true, it's not really even a science yet, and it doesn't look like it has the prospect of really becoming a science anytime in the near future, if at all. The biblical creation, which is contained in the first chapter of Genesis, we've seen to have very strong scientific support, indicating that it is the explanation for life on Earth, that a supreme being that we call God created not only the universe and the Earth, but created individual species of animals over the geologic time that we believe, or the cosmological time that we have, understanding of in terms of the cosmic clock. But that is all consistent with Genesis creation. In part 3, we saw that Adam and Eve and Noah, at least the time at which they lived, has been pinpointed by or estimated by scientific genetic tests, where we looked at male Y chromosomes and female mitochondrial DNA and extrapolated back to when the first human beings lived, and also found, as a glitch in that data, we found the presence of Noah quite likely presented at about 65,000 years ago. We also, in the last part here, in the last fourth part, we saw that lifetimes as long as 1,000 years have a genetic explanation in terms of telomeres and this enzyme we call telomerase. The earlier human beings that were on Earth from the time of Adam and Eve to the time of Noah and beyond are reported to have lived over 900 or around 1,000 years in lifetime, and that is certainly possible scientifically from what we know now about the telomerase and the fact that we could turn that telomerase gene on and have a lot longer lifetimes. The last thing we looked at was the human races and how they resulted from adaptations, an explanation in terms of vitamin D. When people live in different locations on Earth, they need a different quantity of sunlight to make just the right amount of vitamin D. The overall result, I think, of these 4 parts is that biblical creation is the explanation for life on Earth. There are 2 explanations, evolution theory and biblical creation history, and it has been, hopefully you've seen from all the information I've presented here, that biblical creation history is the explanation for life on Earth. It has all of the historical reports, but it has the scientific backing to suggest that this is really what happened here on Earth. There has been no evidence found to the contrary, and in fact, with evolution theory, a lot of contrary evidence has been found which shows that evolution is not true. Now the future, certainly with time here, as more research goes on and more results are found, I think pretty likely that biblical creation will remain the explanation for life. We'll just understand it in more detail, but it will be the explanation. There will be more scientific support, other areas of science where people will have looked at their area of science and said, hey, look, this supports this shown in the Bible in chapter 22 of Genesis or someplace. They will find something that will support that. I think quite likely this genetic extrapolation to find the most recent common ancestor is in a developing area. It's been going on for a while, but I think over time we'll get a better estimates for the time at which these happen, but I don't think it will change drastically. We're probably also going to find more complex organs which will further cause evolution theory to be untrue. More things we will find in living beings that could just not have developed very slowly over time and still had utility during their partial development stages. I think we're going to find that there will be more missing links found, and they will be reported with great fanfare, but shortly thereafter they will be discarded as false. They've been searching for these for 150 years, and 8 or 10 have found of great repute, but they have been found to be false, and I think that will continue. And my dearest hope is that sometime in the future, evolutionists will apply scientific method and discard evolution theory once and for all, or drastically modify it into something that I think would end up being very much like biblical creation history. In any event, that's what I have concluded in these 4 parts. I hope you've enjoyed it. You can hopefully have downloaded some of the video handout material so that you can see exactly what these slides contained. Thank you again.